Photosynthesis, a process that fuels life on Earth, provides energy that sustains the ecosystem, and gives us oxygen to breathe. All of it by converting sunlight, water, and carbon dioxide into glucose and oxygen. And most of the plants on Earth are C3 plants that uses a key enzyme named Rubisco that helps to convert carbon dioxide into glucose. And even though this photosynthesis sounds like a perfect equation, it has a flaw. So today we are going to talk about how plants have evolved itself into surviving the most harsh environments that nature throws at them, where we will see what these C3, C4 and CAM plants actually are. And if you want to learn more about photosynthesis in detail, then I have a whole video on how this photosynthesis actually works. You can check it out later. But for now, let's get back to the video. See, approximately 85% plants on earth like wheat, rice, soybean, etc. follows C3 pathway of photosynthesis, where a key enzyme named Rubisco grabs CO2 and attaches it to a 5-carbon molecule named RUBP. But the formed molecule is unstable, due to which it breaks almost instantly into two different 3-carbon molecules named 3-phosphoglycerate, and hence the name C3 plants. Now, this 3-PGA uses 9 ATP and 6 NADPH to convert itself into a different 3-carbon molecule named 1G3P, glyceraldehyde 3-phosphate. But out of every 6 G3P, 5 gets recycled back into RUBP to do the process again, and two different G3P combines to form a 6-carbon sugar named glucose, C6H12O6. And this Rubisco works perfectly fine until the temperature is normal. But when temperature rises to a more hot environment, plants closes its stomata to prevent losing water from inside the leaf, and due to which new CO2 can't enter the leaf. But the process of producing oxygen keeps happening because sunlight is there. So inside the leaf, the concentration of oxygen rises and carbon dioxide decreases, due to which Rubisco, instead of grabbing carbon dioxide, starts grabbing oxygen, leading to a formation of two different compounds. First, that same 3-PGA, and second, a new useless compound named 2-phosphoglycolate, 2-PGA. And plants can't let 2-PGA to get accumulated, so it uses a complicated pathway called photorespiration, and its aim is to recycle 2-PGA back into a usable form, but it takes a lot of energy. See, at first a phosphate group is removed from 2-PGA, resulting to a compound named glycolate. Now, this glycolate is still useless to Calvin cycle, so it's exported into peroxisome, another cellular component that breaks down molecules by using oxygen. And here, glycolate undergoes oxidation to form glyoxylate, and this process releases H2O2 as a byproduct, which is a highly reactive and toxic molecule. So to prevent cellular damage, peroxisomes contain an enzyme that rapidly breaks down this H2O2 into water and oxygen. Now glyoxylate is converted into amino acid named glycine through transamination reaction, and two of such glycine are combined to form one molecule named serine, along with the release of CO2 and ammonia NH3. And this is where photorespiration becomes especially wasteful, because the CO2 release represents a direct loss of fixed carbon, where instead of contributing to the production of sugar, CO2 eventually gets out of the cell into the atmosphere, unless recaptured by Rubisco again. And this is a wasteful part because plant had already fixed that carbon once before, but now it's lost back as CO2. Also, ammonia produced is toxic and must be quickly reassimilated into amino acids, and this reassimilation requires energy, costing the plant additional ATP through the GS-GOGAT pathway. Also, that serine does not stay in the mitochondrion. It is transported back into peroxisome, where serine is converted into hydroxypyruvate, which is then reduced to form glycerate. Finally, glycerate is settled back into the chloroplast, but before it can re-enter the Calvin cycle, it must be phosphorylated, meaning another ATP is consumed to convert glycerate into 3-phosphoglycerate, 3-PGA. And at this point, the plant has successfully salvaged carbon from 2-PGA, allowing it to rejoin the Calvin cycle to produce glucose. And this is the actual reason why C3 plants had to evolve itself, because it's wasting too much ATP, time, and enzymes into recycling stuff it doesn't need to at normal temperature. And hence, a new type of plants was formed with a more efficient pathway of photosynthesis named C4 plants. So, these C4 plants was probably first evolved around 30 to 35 million years ago, which are mainly found in tropical and warm temperature regions. And these plants at first uses an enzyme named carbonic anhydrase Ca to combine CO2 with water to form bicarbonate HCO3 negative inside the mesophyll cells. And this matters because instead of directly using Rubisco that sometimes starts grabbing oxygen molecule instead of carbon dioxide at higher temperatures, C4 plants at first uses an enzyme named PEP carboxylase for carbon fixation, and this PEP carboxylase not only ignores oxygen molecule but also does not directly react with CO2. So instead it binds to substrate bicarbonate HCO3 negative and phosphoenol pyruvate PEP, a 3 carbon molecule, together into a 4 carbon molecule named oxaloacetate OAA, hence the name C4 plants. Now, this freshly made oxaloacetate is not stable, so it's quickly converted into malate using malate dehydrogenase, or it can also be converted to aspartate using aspartate aminotransferase AAT, depending on what species of C4 plants it is. Either way, we now have a stable 4-carbon acid. Now, this 4-carbon acid moves from mesophyll cells to bundle sheath cells through a cytoplasmic bridge named plasmodesmata. Now, once inside the bundle sheath cells, this 4-carbon acid is cracked open, where malate can be decarboxylated by NADP malic enzyme or NADP-ME to produce CO2, pyruvate, and NADPH. Now, here this NADPH lands precisely where it needs to be for Calvin cycle. Or if it's aspartate instead of malate, then this aspartate can also be rerouted and decarboxylated by other enzymes like PEP carboxykinase PEPK to produce CO2 and PEP, again depending on what plant it is. Anyway, no matter what path it takes, effect is the same. More CO2 is produced right next to Rubisco for its carboxylation in the Calvin cycle to produce glucose, and this CO2 rich microzone is the whole point of C4. Now, after all this, the cycle resets and pyruvate is sent back to mesophyll, where it gets converted back to PEP using PPDK, pyruvate phosphate dekinase, at a notable energetic price. Now, this regenerated PEP is ready to repeat the cycle. Now, contrary of what C4 plants did, doing photosynthesis in a different place like inside bundle sheath cells instead of mesophyll, CAM plants, on the other hand, separate CO2 uptake and Rubisco carboxylation across different times. And let me tell you exactly what I mean. See, at extreme heat environments like deserts, these CAM plants distribute its process of Calvin cycle into 2 time periods, day and night. And the process of converting CO2 into glucose is the same, like absolutely same as C4 plants. The only difference is the time. At night, CO2 is fixed by PEP carboxylase, PEP-C, into 4-carbon acid named malate, stored in the vacuole as malic acid. And at daytime, plants closes its stomata in high temperature environment to prevent loss of water. But now at this daytime, it doesn't need to worry about photosynthesis not happening, because now that stored malic acid from the night process is decarboxylated by one or more enzymes like NADP malic enzyme or PEP carboxykinase PEPK to release a high local CO2 supply right where Rubisco runs the Calvin cycle to produce glucose. So this process is basically the same as C4 plants, but instead of doing it on another place inside the leaf, CAM plants distributed the work in 2 different time frames as day and night processes based on the requirements for Calvin cycle. And here are some extra info to remember, which is that C3 plants are approximately at 5% of all plants on Earth. And this is the beginning of where problem of high temperature and Rubisco starting to grab oxygen leading to photorespiration starts. and C4 plants around 3% and CAM plants around 7% are two ways of nature evolving itself to solve the issue created in the C3 plants. So C3 plants is not a part of solution. It's the beginning of where problem starts, and C4 and CAM plants are alternate solutions of that problem. Anyway, now you know exactly what these C3, C4 and CAM plants actually are. So if you enjoyed the video, then make sure to give it a like. And for more such videos, don't forget to subscribe the channel. And I'll see you back in the next video. Till then, tata, bye bye.